哎，杨老师，杨老师，你帮我看看为什么我的类器官长得又少又慢啊？
其实构建类器官的话需要密集的，就你现在看一下我操作步骤吧。
好呀。
Step-by-step guide to culturing organoids Part 1. Experiment preparation. Spray alcohol on the work surface and wipe it thoroughly with gauze of the biological safety cabinet. Spray alcohol on the instrument kit and place it in the BSC. The instrument kit includes fine straight scissors, fine curved scissors, straight forceps, and curved forceps, all of which must be autoclaved and used within their validity period. Similarly, sterilize all other consumables and reagents required for the experiment with alcohol and place in the BSC. If you are interested in our products, please visit our website www.amymed.com. Step 1. Sample processing. Dispense an adequate amount of DPBSPS into 3 culture dishes and set aside. Note: prepare the DPBSPS with 2% triple antibiotics and percolate at 2 degrees Celsius to 8 degrees Celsius. Open the sample bag. Tissue sample is to be placed in a storage tube filled with Masterame tissue preservation medium and kept at 2 degrees Celsius to 8 degrees Celsius. The sample should be transported to the laboratory within 24 hours for processing. Pour the contents from the sample storage tube into a culture dish. Retain a sufficient volume of tissue preservation medium in a 1.5 milliliters Eppendorf tube for gram staining. Label accordingly. Observe under a microscope to check if there is any cells leaking. Take photos for recording. Place the culture dish of the biological safety cabinet and take photographs from directly above. After photographing, promptly return the tissue sample to the BSC. Wash the tissue sample. Use forceps and scissors to remove any impurities such as adipose tissue, burn tissue, and blood clots. Handle gently and smoothly. Transfer the tissue sample to a second culture dish containing washing solution and continue washing. Repeat the washing process. Place the clean tissue sample in a 1.5 milliliters Eppendorf tube for gram staining. Add 200 microliter of Masterame tissue dissociation medium 1. Cut the tissue sample with straight scissors into pieces between 0.5 cubic millimeter and 1 cubic millimeter in size until the tissue pieces look uniform and mushy. Prepare a new culture dish. Wipe off excess alcohol with gauze. Rinse a Pasteur pipette with a prepared 0.5% BSA solution. Then use it to transfer a small amount of the minced sample into a culture dish and dilute with a small amount of washing solution. Observe the tissue sample under a microscope and take photographs. Note: carefully check for cell leakage, the presence of a significant number of individual cells or cell clusters under the microscope indicates that the mincing was performed correctly. Step 2. Dissociation. Collect the minced tissue. Transfer the minced tissue from the Eppendorf tube to a centrifuge tube containing 4 ml of Masterame tissue dissociation medium 1. Rinse the Eppendorf tube and Pasteur pipette with Masterame tissue dissociation medium 1 to ensure all tissue is collected. If fibrosis is severe, add 500 microliters of Masterame tissue dissociation medium to the tube. Incubate the tube at 37 degrees Celsius with shaking to facilitate dissociation. Monitor the dissociation process under microscope and adjust the time according to the extent of cell leakage observed. Remove the tube from the incubator. Transfer a small amount of the dissociation medium to a culture dish for observation. Examine and photograph the tissue under a microscope to assess the efficiency of the dissociation process. Collect the dissociation medium from the culture dish. Terminate the dissociation by adding 8 ml of DPBSPS to the medium. The dissociation process is complete. Subscribe to our channel for more updates and stay tuned for the next episode.